So, the title of my talk today is Nitrogen Fixing Bacteria in Wheat-Based Cropping Systems and their association with soil health. So, nitrogen, uh, before that, I'll go over the outline of my presentation. So, I will talk first about the nitrogen fixation and specifically about the biological nitrogen fixation and the enzyme nitrogenase reductase, which is responsible for carrying that out. Then NIFH gene, which is a biomarker of nitrogen fixation. And then I'll talk about the effect of different tillage systems, conventional versus no-till, on the structure and composition of nitrogen fixing bacterial communities. Then the correlation between the nitrogen fixing bacteria with soil characteristics and the biological soil health, and then the key highlights or the conclusions. So nitrogen, the air that we breathe in is 78% nitrogen, but most of this is bound up as dinitrogen gas, which has this strong triple covalent bond. And this bond is pretty hard to break. So that's why most of the nitrogen is inaccessible for use by plants. And the processes that reduce this or convert this dinitrogen gas into usable nitrogenous compounds is called as nitrogen fixation. And there are currently three processes known that can carry that out. First is lightning, which breaks the strong triple bond and convert the dinitrogen gas into other nitrogenous compounds. And out of the total nitrogen that is fixed globally on annually, 5 million tons is contributed by lightning in the natural ecosystems. Second is biological nitrogen fixation, which is carried out by the specialized bacteria, also called as diazotrophs, which can live as free living bacteria in the soil, or they can live in symbiotic relationship with the root nodules of the legume crops. And out of the total nitrogen fixed globally, annually, 58 million tons is contributed by biological nitrogen fixation in natural ecosystems and 60 million tons in agricultural ecosystems. And the third process is the industrial process called as Haber-Bosch process, which uses very high temperatures and pressures to combine the atmospheric nitrogen and hydrogen to convert it into ammonia. And it contributes 120 million tons of nitrogen in agricultural ecosystems. So currently, a substantial amount of nitrogen is supplied as synthetic fertilizer in wheat cropping systems and other crops as well. So if we want to reduce the reliance on the synthetic fertilizer, which has certain negative aspects and challenges, the key for that is the biological nitrogen fixation, which is carried out by the specialized bacteria. And this enzyme, amazing enzyme called as nitrogenase reductase, is the only enzyme known to reduce the dinitrogen into ammonia at room temperature. And there are different forms of this enzyme, and it uses multiple metal ions as cofactors. And there are three forms, mainly iron-only form, iron vanadium form, and iron molybdenum form. And here is the picture of this enzyme with two components of the protein. The component one is the nitrogenase component, and the component two is the reductase component. And this reductase component is encoded by this highly conserved gene NIFH, which is used as a biomarker of the nitrogen fixation. And this enzyme is functional at anaerobic conditions, which means that it is inhibited by the presence of the oxygen. So we recently studied the diversity of the NIFH gene from Cook Farm, which is a long-term agroecosystem research network based on uh, uh, wheat-based cropping system, and it's managed under both conventional till and no-till systems. So the soil samples were collected from the Cook Farm, and then they were analyzed for the NIFH gene diversity and the bacterial community structures. So the hypothesis for the study was how the tillage systems, conventional and no-till, impacts the composition and diversity of nitrogen-fixing microbial communities, and how the relative abundance of various nitrogen-fixing bacteria relates to soil characteristics and soil health score. So here is the first graph of the data. So it's the ordination plot, which is basically showing the differences of the community composition in two tillage systems. So the black dots here are the samples from the conventional site, and the pink triangles are the samples from no-till site. And this ordination plot is made from the multivariate data, and it clusters the samples on the basis of the level of similarity between them. So if the samples are far apart from each other, that means they are less similar, and if they are close, means that they are more similar to each other. And the take-home message from this slide is that the tillage systems, they affect the community composition of the different isotrophs. Then we studied the diversity of these diazotrophs using the diversity indices, two of which are shown here, the observed richness and the Shannon index. Both are indices to study the species diversity. The only difference is that in observed richness, it simply means the total number of species in a community. But Shannon index, on the other hand, takes into account their relative abundance. So if I have to give you example, so if you observe a forest and you observe 10 birds there, it means that that forest has a species richness of 10. But on the other hand, Shannon index, as it takes into account the relative abundance, and if one community has many species, but those species are evenly distributed as compared to the other community, which has equal number of communities, but they are contributed by just one major species. So for this particular data set, we observed significantly more richness in the conventional site as compared to the till site, but the Shannon index was non-significant, or it was similar between the conventional and no-till sites. And then we studied the community structure and the impact of tillage systems on the diazotroph community structure. And here is a stacked bar plot showing the different bacterial classes that we observed. And in both no-till and conventional, we observed numerous bacterial classes, but they were somewhat distinct between the tillage systems. And the most abundant phylum was Proteobacteria, and the most abundant classes were alpha Proteobacteria and beta Proteobacteria. So both of these bacteria are a class of gram-negative bacteria, and which are involved in the nitrogen fixation as well. Then talking about the community structure in the for the bacterial families and the effect of tillage systems, we here's the plot showing the top 20 abundant bacterial families. On left, it's in the no-till, and the right graph is for the conventional. So the different colors here represent the different bacterial families. The x-axis is the different samples, and y-axis is their relative abundance. And the top three abundant bacterial families in no-till were Bradyrhizobiaceae, Rhodocyclaceae, and Rhodospiralaceae, whereas in conventional, the top three abundant families were Spirochaetaceae, Rhodospiralaceae, and Methylocystaceae. So the take-home message from this slide is that there were differences between the enrichment of the different families in the different tillage systems. Then this again similar stack bar plot, but here it's showing the different bacterial genera and the effect of the tillage system. So for no-till, the top three abundant genus were Bradyrhizobium, Azospira, and Azospirillum. And for conventional, it was Treponema, Azospirillum, and Pleomorphomonas. So as we observed on a numerous amount of bacterial nitrogen-fixing bacteria, so we wanted to study how the relative abundance of these bacteria associates or correlates with the different soil characteristics and the soil health measurements. So we use the a Haney soil health score, which is calculated by this equation here, which has different components to it. It uses the one-day respired CO2, water extractable organic carbon, water extractable organic nitrogen, and the water extractable organic carbon to nitrogen ratio. So what all these different components means? So one-day respired CO2 means like how much CO2 is released from the soil, which is an indicator of how much active or how much biological activity is in there in the soil. Then water extractable organic carbon is again is the amount of the organic carbon in there. So this equation doesn't count the organic matter in it, but it does account for the organic carbon, because organic matter, you can think it like that. So organic matter is home for the different bacteria, whereas the water extractable organic carbon is the food for the bacteria, right? So they use the organic carbon uh, for their activity. And then the water extractable organic nitrogen is also required for the bacterial activity. And then the soils that have more even carbon to nitrogen ratio, they have better scores. So all these components are added up in, account for the total health score. So we used soil health score and other soil characteristics like plant average nitrogen, the plant available nitrates and ammonia, which are calculated by the plant root simulator probes, and organic matter percentage to see how they correlates with the different bacterial families. So here are the heat maps of the correlations. Left is on the no-till and right is conventional. So in conventional, out of the total 20 bacterial families, 9 showed positive correlation with soil health, with the strongest correlation was observed with this bacterial family Sphingomonidaceae, and others show moderate positive correlation. And also numerous negative correlations were also observed. So if the color is more red in each box here, that means that particular bacterial family has more positive correlation, and the whiter the color means the negative correlation. So we observed 9 out of the 20 bacterial families which showed strong to moderate positive correlation with soil health, and also the same family showed positive correlation with the organic matter. There were some bacterial families which were negatively correlated with the soil health and organic matter as well. And surprisingly, the top 3 abundant bacterial families in the conventional, uh, uh, marked by these yellow boxes and yellow arrows here, the Spirocatiaceae, Rhodospirillacea, and Methylocystacea, all 3 showed negative correlation with the soil health, which is uh, surprising that like these are the top 3 abundant bacterial families, but they are negatively correlated with soil health. On the other hand, in no-till, the out of the 3 top abundant bacterial families, and the Rhodospirillacea showed positive correlation with soil health. So it's vice versa in both tillage systems. And then we did see numerous correlations for the plant available nitrate and ammonia with different bacterial families. And 5 bacterial families showed positive correlation for plant available nitrate and ammonia. And if we compare the different tillage systems for correlations, we observed more positive correlations in conventional as compared to the no-till system. And in no-till, again, we observed only, as I said, very comparatively less correlations. So only 5 out of the 20 bacterial families showed positive correlation, with Enterobacteriaceae showing the strongest positive correlation. So the take-home message from this slide is that there were numerous positive and negative correlations of the different bacterial families with different soil characteristics and soil health. And we identified the bacterial families which are strongly positively correlated with the soil health. And the conventional management had more positive correlations for different characteristics as compared to the no-till. Similarly, here's the correlation plots for the different bacterial genera with the same soil health and parameter and other characteristics. And similarly, for conventional, we observed 10 out of the 34 bacterial genera showing positive correlation with the soil health, whereas in no-till it was only 7 out of 34. And again, there were more positive correlations in conventional system as compared to the no-till. And as I mentioned earlier, the top 3 abundant bacterial genera in conventional again showed negative correlation with the soil health. But in no-till, one of the top abundant genera, which is Azospirillum, showed positive correlation with no-till. So this is another slide showing the correlation of the total soil nitrogen with the observed OTUs. So OTU is it means operational taxonomic unit. It's basically the grouping of the different bacteria on the basis of the sequence similarity they have. So this graph is showing that as soil nitrogen increases, the number of OTUs decrease, or which means that they are inversely related to the soil nitrogen. So the key message or highlights of the study are that tillage systems, conventional versus no-till, they affect the relative abundance and composition of microbial communities. And although the relative although the Shannon index was non-significant, but we did observe higher richness of species in conventional as compared to no-till. Then we identified numerous significant positive correlations of different bacterial families with soil health score and other soil characteristics, and most more positive correlations for bacterial families with soil characteristics in conventional tillage. And on the contrary, many families, bacterial families and genera also showed no and negative correlation with soil characteristics and soil health. And then we did observe an inverse relationship between the observed OTUs and the soil nitrogen. Uh, Thank you, and I'm happy to take any questions.